resistance in grapes that we are working from say 10 years, and we have analyzed the resistant genes, microRNAs, LNCRNAs, and also doing gene overexpression, and we will do editing to produce grapes resistant to fungal diseases. So then we have another improvement of Solanum tuberosum for nutritional improvement and for production of true potato seeds. So this I will talk you tomorrow how we have done the editing in the tobacco, mainly the work done by Hani and Aina. So coming to these medicinal plants, in India's culture, we know that these plants, they are very integral part of our civilization, and we have like Unani, Indian and European and Mediterranean cultures that are using these herbal plants for their medicinal requirements. And as per WHO estimate, roughly 80% of the world population, it depends upon the medicinal plants for the treatment of diseases, and we have more than 21,000 plant species that are potential to be used as medicinal plants. And in the developed countries such as United States, there are almost 25% of the drugs that are direct obtained from the plants, or they are the derivatives of plants based drugs, like we have artemisinin or quinone for the treatment of malaria in many countries. They are directly derived from the plants. And in countries like India and China, we are more reliable on these medicinal plants for our daily needs. For example, 80% or more than that, they have been using these medicinal plants. And also, this treatment is considered to be safe with minimal or no side effect. Okay, eating a plant doesn't have any side effect. So this is considered to be very safe. Apart from these medicinal properties, it also involved in like human civilizations development. So many civilizations across the world, they worship one or other plants, like in our Hindu culture, we worship many plants. In our Sikh religion also, we worship many plants that are very holy to us. So every civilization, every race, okay, they have their own sacred plants that they worship and use for their benefits. So for example, we have a ginger, green tea, walnut, pepper, turmeric. They have therapeutic value. We can use them in natural dyes, pest control, food, perfume, tea, okay, and so on. And they have antimicrobial, antifungal, anti-pest properties, and we know that we can burn leaves of neem, okay, lemongrass to keep these flies away from the homes and offices. And they are used in many treatments. And over the last 2 decades, the use of these medicinal plants, they have been increased in India because of what? Because of Ramdev ji? Yeah. So when Baba Ramdev ji came into picture, then people of India, they suddenly started believing more and more in the herbal plants. But it's not, he has credit to make it popular among the people. But before him, so we have Ayurveda, Charak Samhita, everything was there. But we were more going towards the allopathy and other things. But now, for example, if you ask me personally, I prefer to go first to herbal treatment. Then if it's not curable, then I go to doctor. Everyone tries to avoid doctor as much as they can. Okay. Then if we talk about the medicinal plants, then that we have 2 kinds of metabolites, primary metabolism and secondary metabolism. So medicinal plants has high amount of the secondary metabolites, particularly belonging to some group or category. So their amount is very high in some cases, like it's sometimes more than 10% of the dry weight of the organism. And they have extremely complex structures, and they have a very unique role in plants, like they protect the plants from pathogens, from fungal infections, bacterial infections, protect the plants from UV, and also they are nurturing the ecosystem around the environment. So that is the main benefits of secondary metabolites, that they are not used by the plants for himself or itself, but they are produced for their nearby relatives in the ecosystem also. Maybe the plants or any other association, like for example mycorrhizas or lichens, they are there. They are sensing these compounds to make associations with the plants. So with high amount, that we say that they must be doing something, and now we know that what they are doing. Okay, and then natural products, they are the basis of a big percentage of drugs that are commercially available in the market, like we have artemisinin and quinine and many more are there. So I normally give talks on the secondary metabolism in our MSc courses, where I tell that what kind of drugs they are there. But these compounds, they are produced mainly from the pathway like shikimate pathway, then we have alkaloid biosynthetic pathway, then we have terpenoid biosynthetic pathway. These are the three main pathways: phenylalanine, phenylpropanoid, flavonoid, alkaloid and terpenoid, that are producing more than 100,000 different kind of compounds that are used in many different applications. Okay. So don't worry, we will give you slides. Okay. We decided to give you all the slides, so don't worry about that. Don't click pictures or anything. So India is a one of the large country, okay, and it has 15 agroclimatic zones. So every 100 kilometer you will see the change in the vegetation and other things. And we have roughly 17 to 18,000 species of flowering plants, and 6 to 7,000 they are estimated to have medicinal uses. Like they are systematically documented in Ayurveda, Siddha, Unani and homeopathy medicine system. And we are trading like more than 100 metric tons of plants. Now we have many companies like I visited Kapiva, Nakama Ayurveda. They are directly procuring the raw material and then processing it and selling it at very high price, because they are maintaining the quality standards. So they are also provide livelihood and health security to a large segment of Indian population. So there are many people living in Himachal and Uttarakhand. So you ask them to bring plant, they charge money from you, and they can bring you plants that we try to use them, but luckily we didn't get them, because some plants, they are extinct and don't know or endangered, and they know from where they can get it, and they bring it, but they never tell us the source from where they get the plants. Okay, so that is something that we need to stop this kind of illegal trade of plants, but at the other hand they are required, so they have to promoted and protected for medicinal uses. Now we have made a separate Ayush industry, Ayush department of India. It's taking care of all the medicinal plants that has been produced in India, and they are increasing the trade of herbal up to $7 trillion by 2050 or more than that. So in my lab, our research is focused on secondary metabolism in medicinal plants, wherein we are identifying the structural genes of secondary metabolic pathways such as flavonoids, alkaloids and terpenoids. And for these we are using high throughput transcriptome sequencing. Then we also try to find out the microRNA targeting those genes. So we are doing small RNA sequencing. Then we also do analysis that how these small RNAs, they are regulating the secondary metabolism. Then gene expression analysis, and in our lab we are working on many plants, but here I will show the results of chlorophytum and Sasauria lapa. So chlorophytum is commonly known as Safed Musli. It's ranked 6th among the 28 medicinal priority medicinal plants selected by the Medicinal Plant Board of India. So its demand is 35,000 tons per year, but supply is only 5,000 tons per year. It's grown majority in the Madhya Pradesh, Rajasthan. Okay. But the people of Punjab and Haryana, they are also growing it, because the cost of this plant is very high. It's sold in the market like dried roots of this plant. These are sold as between rupees 2,000 to 3,000 per kg. Okay, and it doesn't require any specific requirement. It's a very hardy plant. You can culture it and it can be grown anywhere. It needs only sandy or loamy soils. So it's used in phytopharmaceutical and nutraceutical products because of its immunomodulatory and many other properties. So it has many references in the Ayurveda classic like Charak Samhita, and it has been used in Ayurvedic system of India from the 11th century, so it is documented. Mainly its properties are aphrodisiac agent and vitalizers, and its dried roots, they are used in more than 100 different herbal formulations. So already the formulation derived from its dried roots, they are available in the market, and it is considered to be as a herbal Viagra. Okay, so it not only it treats the sexual related diseases, but it also promotes immunomodulatory activities, and overall health of the person increases when they take this Safed Musli. So if you combine it with Vidania somnifera and Amla, and it makes your body resistant against diseases, sex-related disease, and delay menopause. Okay. Now, dried roots of these plants, they are mainly consumed, and the key ingredients are carbohydrates, proteins, and that's normal. But one thing, that saponins are 10 to 20%. That's very high number of the saponins they are present there. So it has very high amount of saponins, so that's why saponins are considered to be responsible for the medicinal properties of this plant. And among all the species, Chlorophytum borevelinum has highest saponin contents among different species, and hence we selected this plant for our study. So what we did, we analyzed the pathway of formation of steroidal saponins. So it is formed through terpenoid biosynthetic pathway starting from mevalonate pathway and so we have two pathways operating for formation of terpenoids. It's MBA pathway and MEP pathway. So one is operated in the cytoplasm and another in the plastids. So when we started working in this plant, none of the genes like mentioned here, they were known from this plant. In fact, no one was working on this plant on exploring the molecular biology applications or gene clonings. Only tissue culture related literature was available. So we started working on these plants on the molecular aspects, and then 3-4 students, they did PhD on all these objectives. So first one, identification of genes involved in saponin biosynthesis using transcriptome sequencing. At that time we had sequencing with short reads available up to 50 bases. So then we did raise PCR. Then identifying microRNA regulating saponin biosynthesis by small RNA sequencing and degradome sequencing. Then we did gene expression analysis, and after that we did functional characterization of pathway genes for enhancement of saponin production by hairy root induction and gene over expression. So these two students, Shikha and Sunil, they worked on leaf and root transcriptome in this plant, and when we started this, the Illumina was not up to 300 base pair read like they are giving nowadays. So they were producing only 50 cycles reads, so 50 base pair in length. So then because that technology was not available in India, so we sent our samples to USA for sequencing, and they produced this amount of single end reads, and then we filtered the data that we discussed in the morning, how to filter the data. Then we assembled the data that was having a Phred score more than 20, and after this analysis we were left with this much number of high quality reads. Then we assembled this transcriptome data using because we used our systems in-house facilities, so we used SOAP Novo and K-Marsage was 23, like I said in the morning, that overlapping. So we overlapped 23 bases. Okay, and then we did clustering. Nowadays people don't use this, like I told you in the morning, that this may lead to elimination of the isoforms or splice variants from your clusters. So then we did TGICL and CAP3, and after this we were left with more than 100,000 transcripts that we further used for annotation by BLAST. So after doing BLAST and all other analysis like BLAST, KEGG and GO mapping, so we were able to identify all the genes of the pathway, like the numbers written in the red along with the this gene, they are the number of transcripts that we identified for these particular genes. So multiple transcripts encoding almost all known enzymes involved in MVA and MEP pathway and saponin biosynthetic pathway were identified. So saponin formation starts from here, squalene, squalene epoxidase. So two types of saponins are there, steroidal saponins and triterpenoid saponins. So you can see we have very high number of cycloartenol synthase transcript compared to beta-myrin synthase. So we assume that we have high amount of steroidal saponin formation in this plant. That we tested with HPLC also, but I will not show that results. So copy number of cycloartenol synthase was higher as compared to beta-myrin synthase and dema-myrin synthase. And along with these saponins, we also found excuse me, transcripts involved in flavonoid and alkaloid biosynthesis pathway. So because since my PhD, I was very much interested in flavonoid biosynthetic pathway. So I found all the transcripts involved in flavonoid biosynthetic pathway in our data, and also some of the genes of alkaloid, because this chlorophytum has some alkaloids present in it, and then we published that into PLOS One and other journals. Then because at that time we chose 50 base pair single end reads, so we got short transcripts, and then we did race experiments to clone the full length genes of this pathway. So 7 genes we have cloned full length, and then we characterized them, some of them by doing recombinant protein expression and by enzyme assays. For example, we did assay for SQS and SE for sure that they are converting squalene into squalene epoxidase and other things. And then also at that time we did semi-quantitative gene expression because we didn't have the real time at that time. So we saw that the initial genes of the pathway, they were expressing high in the leaves, and the later genes they were expressing high in the roots, means modification and storage, because roots are the primary part of this plant that are used for medicinal properties, because it has very high amount of saponins. And then with our data we proved that the initial steps of the pathway, they are done in the roots, because the pathway is mainly updated in the plastids. So plastid is only present in the leaves, and after making that, the metabolites, they are transported to the roots for their final modification by this GTs, glucoside transferases, and CYPs, cytochrome P450 oxidases. So they were modified and stored in the roots. So leaves were the primary synthetic part of the plant, and roots were the modification and storage part of the plants. And then we did identification of microRNAs regulating the saponin biosynthesis. So for this we did small RNA sequencing, and after filtering the data, we identified 251 conserved microRNAs belonging to 38 different microRNA families, and also we have identified 300 novel microRNAs, and we have also found putative targets of these genes involved in saponin biosynthesis alone by using some top wells, as also by degradome sequencing. Okay, so in this we did again smaller end sequencing using HiSeq. This we done in India because at that time this technology came to India. Then read quality check, adapter removal of non-coding RNAs like tRNA, rRNAs. Then we aligned our reads with the MIR base for known microRNA identification. Then predicted the novel microRNA and their secondary structures. And then we predicted the targets and analyzed the targets with GO and KEGG pathway mapping. So this is the known microRNA families. Their number, they are written in here, how many transcripts or how many reads for these we got. Then also we calculate the number of each known microRNA families and their length. Okay. Then we also identified some novel microRNAs, and we confirmed these novel microRNAs by making these hairpin kind of structures. And we typically found these 5 novel microRNAs that were regulating the saponin biosynthesis. Then after this, to confirm their present, we tried to clone these and sequence them, and then confirm that they are there. Okay. Then after doing all these analyses, we were able to show that we have many microRNAs that were regulating these genes. These genes, they are typically producing saponins in this plant. Okay. So squalene epoxidase or squalene synthase, squalene monoxidase, they are all the important genes that are producing the saponins in this plant, and we found that microRNA targeting this. And after this KEGG and GO mapping, we were able to locate the targets of these known microRNAs with the known genes involved in the microRNA and gene interaction network. And then we studied after doing this analysis, we studied the real-time PCR to check the relative expression level of microRNAs and their targets, and we found that when we have low expression of microRNA, then their corresponding gene expression is high, and when we have high expression of microRNA, then their corresponding gene expression is low. Then somehow we were able to correlate the expression of the microRNA with their target genes. And after completing this, our task was to increase the saponin production by hairy root induction and gene overexpression. So another student, he started working on this. So because this chlorophytum is a monocot plant, and it's not very easy to transform this chlorophytum. So he developed the protocol for transformation of this aerobic plant by agrobacterium rhizogenes. Then he constructed vectors for SQS and SC and did transformation and did analysis of saponin contents in the hairy root transformed plants and non-transformed plants. So these are some of the steps that he performed to make micropropagation protocol and also transformation protocol using the stem disk. Then we had 2 copies of SE genes, 2 distinct SE genes were present. So we, based on our analysis, we chosen one gene and try to establish its role in saponin production. And for this, we chosen tobacco to transform first, and then later on we shifted to chlorophytum. So in tobacco, we used Picambia vectors and PRI, 2 distinct vectors, and transformed them, and you can see the transformed tobacco with high amount of hairy root produced on them. And when we analyzed them, you can see the transgenic plants, written here T1, T2, T3, T4, T5, and these are the wild type plants. So if you compare these transgenic plants, their height was very, very low. So we did this number of times, and we have distinct lines, all of them expressing SC. They were shorter in size compared to the wild type plant. Somehow their internode was very small. Number of leaves were same when we compared. So this is again the same plant. But here you see this is wild type plant and this is transgenic plant. So somehow it was also regulating the inflorescence number. So we have more than 10 inflorescence per plant compared to 2 to 3 in the tobacco. And this is wild type plant. You can see the leaves. So here the leaves they were like curved, and when you see the transgenic, they have very high number of roots. So we also analyzed the saponin contents. It was high in those plants, but why these plants they were abnormal, that was not we were able to understand. Okay, so normally this kind of results they are not expected when you are changing saponin. It should change the saponins, not the primary physiology of the plant. So we have to check what other kind of changes this gene have done, why the plants of this tobacco, they have reduced height. Otherwise, this will not work. Okay, so we are, I'm looking for a PhD who can do transformation in this chlorophytum so that we can could take this work further. Then another plant we studied is Caesarea lapa. So these two girls, they have already completed their PhD on it, and she was a postdoc in our lab. So this Caesarea lapa is a high altitude plant grown at a height more than 3000 meters. Okay, so this plant is rigid, like its roots, they are very rigid, and roots are again the primary part of this plant that are used for medicinal properties, and it can grow up to height of 1 to 2 meter. The leaves are very broad, okay, and the roots are sturdy with characteristic smell. So people, when they have digestive disorders, they simply take out the root, grind it, and eat it. Flower colors varies from dark purple to black, heads and stalkless, stiff and rounded. In India, it's native to J&K, Himachal Pradesh and Uttarakhand. Normally, you can find good plants at 3500 meter height. And dried roots and root oil, they are the major source of active constituents, both used in traditional and modern medicine. So, if you talk about the principal compounds, sesquiterpenes, they are the active principles of Caesarea lapa and responsible for most of these medicinal properties. And among these STLs, we have costinolide, that is one of the major biologically active compound with known pharmacological applications. So again, the many products from this plant, they are available in the market. So the properties include it relieves pain, improve digestion, treat chest pain, vomiting, diarrhea, dysentery, tenesmus and dysphagia, regulate the immune system and central nervous system, promotes key circulation, anti-inflammatory, anti-cancer, antimicrobial, treat cardiovascular diseases, hepatoprotective effect and anti-cancerous effect. So these are the well documented uses of this plant. Now in this plant, the costinolide biosynthesis again was very poorly understood, so no one was working on this plant again. So luckily we have chosen those plants on which no one was working, so we have independence to work at our own speed, no rush in publication and other things. So again, we did transcriptome sequencing for costinolide biosynthesis, then identification of transcription factors regulating costinolide biosynthesis, then promoter cloning and their interaction with transcription factors, and identification of microRNA regulating costinolide by small RNA and degradome sequencing. So what we did in this case, we collected the plants from Chitkul. Chitkul is in Kinnaur, Himachal Pradesh. It is called the last village of India. So ahead of this is China. So we isolated RNA from young leaves and roots and sent for sequencing using Illumina. Then we did assembly and annotation of the assembled data, identification and characterization of genes for costinolide biosynthesis and also transcription factors, the annotation based identification of costinolide biosynthetic pathway genes, then differential gene expression, construction of co-expression networks. Then we did promoter cloning by gene walking, in silico analysis of promoter sequences, identification of associated transcription factors, and then yeast 1 hybrid assay to study interaction between promoter and the transcription factors, and qRT-PCR based expression analysis. So this is the plant growing in its native area in the Chitkul, so we are collecting that. The thing was, these plants, they were not able to grow in this region, so we have to process them. Either we bring them in liquid nitrogen directly from that source, or we bring them and they grow for some like 15 days or 1 month, and then we immediately harvest it because they need extremely low night temperature for their survival. That was impossible for us to maintain here. And then we send leaf and root samples for sequencing at get this much number of reads. Uh, so this time we had little bit more money, so we did everything in triplicate and get more amount of data. And then we did basic like filtering, assembly, and then we did blast analysis with a non-rendered database. So did we evaluate distribution, blast text similarity, the number of plants with our transcripts they were matching, and the transcript annotation by several databases we used for analysis. And after this analysis, we were able to find out almost all the genes that were involved in making this costinolytes, starting from HMG coenzyme A reductase to malonate kinase, phosphomalonate kinase, diphosphomalonate kinase, IPP, IDP. So every single gene was there start to the costinolyte synthase. Okay. So we almost identified all the genes of the pathway and then published this paper in Genomics. And then we want to analyze the transcription factors regulating these genes. So we retrieved the transcripts and search against the plant transcription factor database, and we encoded like roughly 30,000 transcripts. They were putatively transcription factors in our transcriptome, and then we characterized the families of these transcription factors according to the plant transcription factor database. Then also we did Go and KEGG pathway mapping, where these transcription factors they are involved in biological processes, cellular components or molecular functions, and then by KEGG we were mapped against plant pathogen interaction, signaling, biosynthesis of secondary metabolites, and many other functions they are involved in. So we were particularly interested in the transcription factors that were interacting with the genes. So we did the co-expression network analysis and found that which are the transcription factors that are co-expressing like our genes of the pathway they are expressing. So we did in silico differential gene expression analysis and found out some of the transcription factors that they were binding to our pathway genes, starting from IPP to costinolyte. And we did this kind of co-expression network analysis using the co-express software, where it tells you that which genes they are co-expressing with your transcription factor and vice versa. So we developed these different modules for co-expression data, and in one of the module we found that we have the pathway genes like GAO, COS, DPD and GAO, where many pathway they are interacting with them and expressing in the similar manner our genes are. So in the end we find out that transcription factors like that were interacting with the pathway genes like GAS1, GAO, COS1 and DPD. Okay, they were this is the primary gene of the pathway, means at the very beginning, and GAS and COS they are the last genes of the pathway, and after this COS we have direct formation of the costinolytes. And then we decided to clone the promoters of DPD and COS, and for this we did genome walking. So we make genome walker library, we isolated the DNA, digested it, ligated adapters, and did PCR to clone the promoter sequences. So after this genome walking, we were able to identify 777 base pair promoter sequence of DPD, and then we analyzed the promoter using the plant care and place database, and we found like there are certain boxes you can see. So these are the elements that regulate the promoters. For example, we have light regulating, cold responsive, GATA box. Along with this, we have certain transcription factor binding site like MYC, W box, MYB. So they were there. So our target was to identify the transcription factors that were binding to our promoters. So similarly, we analyzed the promoter and found many sites that on these sites which kind of transcription factor they are binding, and finally we found 10 different transcription factors. 4 were Vicky, 4 were DOF, and 2 were BHLH. They were binding to the promoter. So finally we identified 10 targets, and similarly we did the same in case of the cause. We cloned the promoter sequence of the cause, analyzed it by plant care and place database, and find out the elements that were responsible for regulation along with the MYB, along with the transcription factors binding to this promoter. And here also you can see we have many sites that were having MYB binding sites. Okay, so in the DPD we have 3 different transcription factors, and here only one transcription factor MYB that was binding to the promoter. So we shortlisted these MYB transcription factor in the cause and 3 in the DPD. Okay, so we selected them for further analysis and want to confirm the association of this transcription factor with the promoter using yeast one hybrid analysis. So what we do in yeast hybrid analysis, you have a one bait vector and another is the prey vector. So you put your bait like promoter in one vector and prey your transcription factor in another vector, and then put into same yeast strain. And if these two interact, then the yeast is able to complete its metabolism and the colonies will grow. If they are not interacting, the pathway will not be complete and yeast will not be able to grow. Okay, so we did all the cloning and vector construction like we do for normal cloning expression, and जो भी आप लोग करते हैं, we do the same thing. And in the end, after making the bait and prey, we found these are the positive and negative controls. So we were able to found that DOF2, BHLH3 and VIRKI2, they were showing some positive associations with the promoter of the this DPD, and then we got colonies here. And similarly in cause, we have MYB2, we identified like 7 MBVs. Out of those 7, MYB2 was able to show the positive regulation, and here we found 4 of this DOF, 2 of BHLH and 4 of VIRKI, and out of this only 111 were showing the interaction with the promoter. So we shortlisted from 30,000 transcription factors to 4 transcription factors which were showing interaction with the promoters of these 2 genes, DPD and COS. So we got very good results of yeast 1 hybrid assay. So we identified that DOF2, BHLH3 and VIRKI2, they are interacting with the DPD promoter, and MYB transcription factor were interacting with the COS gene and showing positive interaction results. And so relative gene expression analysis we did in that and showed upregulation of COS as well as its corresponding MYB in the root tissues, and similarly DPD genes and its as interacting transcription factors, they have high expression in the leaf tissues. So the same thing was there, that the primary pathway it operates in the leaves, and the last stages they were covered in the roots. Then after doing this transcriptome analysis and finding out the transcription factors, we further moved to small RNA sequencing and degradome sequencing and network analysis to determine potential mRNA as involved in regulation of costinoid biosynthesis in the Sassoria lapa. So again, what we did here, we did like small RNA sequencing, we sent the samples of leaf and roots, did library construction, sequencing, did fast queue results, read processing, filtering reads, and after filtering the reads, removing tRNAs and rRNAs, we analyzed the reads with the MIR base for identification of novel microRNAs, and with the remainings, we identify with the known microRNAs and the novel microRNA. And also for target prediction, this time we did degradome sequencing, uh, and that maps the cDNA. We created degradome profiles, and we were able to identify the potential mRNA and their targets, regulating the costimulide biosynthesis. This is again some of the steps that we discussed in the morning that you have to do for bioinformatic analysis. For microRNA identification, you can align to the reference genome. If you don't have, you have a MIR base that can characterize the known microRNAs, or you can do cross species like we did in Arabidopsis, Helianthus, Sassoria-Lapa transcriptome, because you don't have genome sequence available either for Safed muesli or for this Sassoria-Lapa, using different tools. And we were able to identify 463 known microRNAs and 594 novel microRNAs for this, and out of this we found 44 unique target core for 12 enzymes in the costimulide biosynthetic pathway. So then again we did basic analysis, the size, lengths of these microRNAs, their expression in leaves and roots, how they are behaving. And then we did degradome sequencing to find out the targets of known microRNAs and novel microRNAs from our data. Then after doing this, we also did networks in which pathway our microRNA, and after compiling of all RNA sequencing, degradome sequencing, and network, found that we have 1 microRNA targets in novel, 25 microRNA targets in the known, and we use, because microRNAs they mostly target the, so we used Arabidopsis transcription to find out the microRNA associations. Associated microRNAs, they were identified for homolog plant, and we used gene and gene microRNA, this interaction network analysis, and make the transcription network. And in the network analysis, we created in their associated transcription RNA and novel microRNA. Of graphs, it's hard to understand, but you can see one microRNA can target many. So similarly here, the many genes, they are being microRNAs taken. So if you see these kind of graphs, that means they are good to understand further. So then enzymes of the pathway, like you the enzymes, costimulide, mevalonate, farneside diphosphate. So the positive interaction with the transcription factors, microRNAs, and analysis, we were able to RNAs regulating the belonging to different categories here. Okay. And we have identified and characterized genes, transcription factors associated with biosynthesis, costimulide in Sassoria-Lapa, then core and differential expression pattern along with a series of were analyzed to successfully reveal 4 transcriptions with the identified promoter regions of COS genes. Then interaction analysis transcription factors and their specific costimulide pathway genes showed 4 positively interacting proteins. Further, the expression studies were performed for genes and transcription factors in the roots and leaf tissues of Sasoriolapa to support our results. And the microRNA genome analysis predicted the known and novel microRNAs in this plant. And because these were the first kind of we are doing, and now many Chinese and other labs in India, they have started working on this plant, but our work was the foundation in finding out the genes and their associated regulatory factors in this plant. So in this time we got funding from like many institutes like DBT, CSIR is there, SCRB is there, and DST, who supported continuously to perform work on these 2 plants. So in the end, thank you very much for your listening. So any question you have, you can ask.